annual holiday party, which we typically do at the end of the semester in December in the gallery with lots of food and friends and family, and we're all packed in there. And under the circumstances, we're not going to be able to do that in the gallery this year. And from talking to people, my sense is there's enough Zoom fatigue so that a party by Zoom is not really a party. So I think we're not going to do that either. But what we're looking into is having a holiday party outside under a tent near VLSB. And I'll send more information about that. It'll likely be between 3 and 5 p.m. So we'll do a little earlier than usual, just because it'll be getting dark around 5. So that's the plan, details to follow. And if you have any suggestions or comments, feel free to send them to Carly or me, but I think that's where we're headed. And then for the MVZ symposium, the annual symposium we do every year in January before classes start, where we have a bunch of short talks, we're planning to go ahead with that and do it in person, and we'll likely do that in the Brazil room. It may be on a different day than usual. We usually do it on the Thursday before classes start, and we're looking at the Wednesday before classes start, just because of availability of the room. Carly, do you want to say anything else about either of those events?
Nope, that covers it.
Okay, super. I'll turn it over to Jim to introduce our speaker today.
All right, great. So yeah, it's my pleasure to introduce a new postdoc in my lab, Simon Scarpetta, who's today's speaker. Many of you have probably already met Simon. He's got a boisterous personality, and he's already been interacting with tons of people around at least this side of the MVZ. Just a little bit of background. Simon grew up in Albuquerque, New Mexico, and he worked in former MVZ postdoc Steve Poe's lab as a high school student. So he got his start with science early, working with the Knolls, and then he went to Stanford University, just down the way, where he worked as an undergraduate in Liz Hadley's lab, so another person with a connection to the MVZ. And while he was there, he actually worked under the sort of direct mentorship of Melissa Kemp, somebody else many of us know here. And in fact, he got his interest in fossils by joining Melissa doing field work in the Caribbean, where they were looking for anolis fossils in cave deposits. And so that motivated his interest in fossils, and he was definitely in a lab that really sort of bridges the neontology, paleontology perspective. And he continued with that tradition when he went off to the University of Texas to do his PhD with Chris Bell, somebody who I think is also familiar to many of us here in the MVZ. And he did his work there on Miocene fossil lizards, but part of his program there was involving both a neontological perspective as well as this sort of paleos perspective. And this really led to him developing a postdoc idea for which he received an NSF postdoc to bridge the neontology, paleontology perspective with respect to iguanian lizard phylogenetics. And iguanian lizards are a huge group. There are about 1,500 species in this clade, so they're one of the most important clades. They're certainly the most interesting clade of squamates. That's my unbiased perspective on that. And Simon proposed to use an exome capture approach, a phylogenomic approach, to study the phylogenetics of this major assemblage, but then to bridge in a much more rich sort of incorporation of fossils to try to be able to do a more appropriate sort of divergence gating sort of analysis for this giant group of lizards. And so I'm super excited about the project. He's hit the ground running already. Simon is in the office that's in the larger office that's next door to mine, not in my side office, but in the next office over. So if you, and he's been in virtually every day. So if you have an interest in meeting him, especially after seeing his talk today, I encourage you to go down and introduce yourself. Simon, take it away.
Awesome. Thanks for the very nice introduction, Jim. Much appreciated. All righty. So which screen did I share? All
think we were seeing your non-opened version. Yeah, we see your presenter view. There you go.
Okay, so everything look good now?
Yep.
All right, sweet. All right, so how's it going, everybody? Again, my name's Simon Scarpetta. I just got my PhD at the University of Texas at Austin, and I'll give a little bit more background about that in a sec. And today I'm going to be talking about a few projects from my PhD where I was thinking about the effects of phylogenetic uncertainty on fossil interpretation for 3 really nice fossil skulls that I was interested in and had the pleasure of studying. So I'm an evolutionary biologist, I would say, but my research program and my interests have led me to try and combine, 3 main classes of data to try and answer questions in evolutionary biology, namely geological data, even though I would say I'm probably not a geologist, but I try, and I've done some geological fieldwork in the past, and then data from fossils, and then data from the modern biota. So, like this Phrynosoma modestum that we've got there, and divergence time analyses and things like that. But my interest in herps is longer than my interest in science, actually. So I've been herping since I was like 2 or 3. Initially, actually, my primary interest was in salamanders, and I still love salamanders to this day. And so my fascination with natural history and going out to different places and herping has really driven a lot of my career, I would say. And it's definitely an important part of what I do to be able to work on the group of animals that I care about, and I've spent a lot of time thinking about. And it's a real pleasure to get to work on herps. So here's a little bit about my research background. So like Steve said, I grew up in Albuquerque, New Mexico, and I was able to start working in Steve Poe's lab when I was in high school as part of a senior project. And so that involved collecting external morphological data for Anolis that ended up going into species descriptions or redescriptions of a few different species of anole, namely Anolis alcopensis from Panama, and then Anolis alvarez del Toroi, which is a cave anole from southeastern Mexico. There's Steve right there in the field with the boa constrictor that we found road cruising. And then there's this really nice Anolis compressicata that I found. It was like moving around at night in Veracruz on the ground, which was pretty fun. And then I went and did my undergrad at Stanford University, where I was in Liz Hadley's lab. But like Jim said, my direct mentor was Melissa Kemp, who's now a professor at the University of Texas at Austin, where I just got my PhD. And she is someone who really elegantly bridges the gap, again, between neontology and paleontology. And so I think from her, that's where my interest in fossils really started to develop. When we went on a trip to the Isle of Guadeloupe, actually an isle called Desirade off the coast of Guadeloupe in the Lesser Antilles. And we were collecting subfossils from cave deposits there, and then also catching the native species of Anolis there, Anolis desirade. And so once my interest in fossils had been developed, I went to the University of Texas at Austin, where I worked with Chris Bell, who is another former MVZ/UCM peer. And so my interest in the fossil record and how we interpret the fossil record and how to put that together with data from the modern biota continued to develop there, as did my interest in alligator lizards and iguanian lizards, which Chris has also been interested in over the course of his career. So I've been interested, and I've studied a lot of different stuff over the past 6 years and before. So namely, taxonomy and morphology of anoles. I've also been interested recently in lizard tooth pathologies and the fossil record of Teid lizards, and fossil record, divergence times, and paleoecology of phrynosomatid lizards, so horny toads and earless lizards and Scarpetta and things like that. And then the phylogeny, biogeography, and fossil record of iguanians. Obviously, there's some overlap between these two. And then the phylogeny and osteology and fossil record of alligator lizards. And so it's these two right here that I'm going to focus on today. But I just wanted to point out that there's a lot of different stuff that I've been interested in, and broad interests. So I'm going to focus on these 3 really nicely preserved fossils. These 2 are alligator lizards, and this one is an iguanian lizard. And so I'm going to talk about, how I went about figuring out what they were and what I learned about interpreting the fossil record more broadly from looking at these, 3 very nicely preserved fossils. And so that's part of the reason I wanted to talk about them, is that they're beautiful fossils, and that's cool to find that in the lizard fossil record. And then, I'm really interested in alligator lizards and iguanian lizards generally. But it's really important to note that in terms of thinking about, how do we think about what fossils are and what interpretations that we can make from them? Alligator lizards and iguanians are 2 really good groups to look at, because there's been quite a few hypotheses and intra-group hypotheses and how everything is related to each other. And so they both provide a good opportunity to kind of get at that. So before I continue, though, I want to point out that looking at beautiful fossils like this is not the reality of most paleoherpetology. So most of the fossils that you look at or that you would find in a collection are going to be more like this. So really scrappy pieces of the dentigerous bones, or sometimes you'll get something like a quadrate like this. And that doesn't mean that you can't do anything with them. So, this fossil and this fossil I actually formally described, but it does limit your ability to interpret the fossils. And so, when I first started my PhD, I had a lot of ideas about meta-analyses of the fossil record of North American lizards that I would like to do. And then when I went in and started realizing what the material record actually looked like and what kind of interpretations I could make with those fossils in the phylogenetic context, that kind of went out the window a bit. And so I got much more focused on, how do we actually do the primary interpretation of fossils in the first place? And so then that's what I spent a lot of my time doing. So I'm going to talk about alligator lizards first, but I first wanted to point out my 2 main collaborators on the project. So the first is David Ledesma, who is my undergraduate mentee for the first few years of my PhD, and then he graduated and is now doing his PhD in the Department of Integrative Biology at UT Austin. So really cool to see him grow up and his development as a scientist. And then, of course, the other collaborator is Chris Bell, who's my advisor, or was my advisor as a PhD student. There we are, both looking very happy on graduation. Okay, so let's talk about alligator lizards a little bit. So alligator lizards, are a group that everyone's probably familiar with because they're found around the Bay Area. There's 2 species, northern and the southern alligator lizard, but they have a pretty broad distribution in the western part of the United States and Western Canada, and then they're also found in Mexico and some parts of Central America. There's around 60 species, so not a crazy amount, but they live in a lot of different kinds of habitats. So here I've got some sort of scrubby desert forest in Nuevo Leon. They also live in cloud forest, namely this group of Bronia. And they live in all sorts of habitats in California, scrubby, mesquite, in the redwoods, you name it, alligator lizards can probably go live there. So in terms of the fossil record, there've been a number of fossils that have been described, mostly from the Cenozoic, but there have been a few hypothesized ones from the Cretaceous as well. But it's been pretty challenging for paleontologists to try and put exact phylogenetic placement on what those fossils are. And so, why has that been the case? Of course, there's been some argument or some multiple hypotheses presented on how different alligator lizards are related to each other. So here's one early one that David Good presented or proposed based on osteology. He's another former MVZer actually. And then here's another one more recently that's based on a few nuclear and mitochondrial markers. And then here's another hypothesis that is based on a much larger set of data. So you can see there's been, some disagreement about what's the most basal genus, and then, whether these alligator lizards, the Bronia and these aspis, are sister to each other or part of the same clade. And I'll get to that in a little bit more detail later, but I just wanted to point out that there's some phylogenetic uncertainty for the group. And, so it makes it difficult to find consistent differences in fossils if you don't know what the natural groupings are. Adding to that is that the skulls of the extant taxa can themselves be difficult to interpret. And that's namely because of the osteoderms that are fused to the skull in most prepared specimens, which can make it hard to see the internal anatomy. And with respect to the phylogenetic problems that I was just talking about, a lot of previous workers on the osteology of alligator lizards actually found that, despite there being, despite these different groups being pretty easy to tell apart when, you're looking at the external morphology, it was surprisingly difficult to tell them apart when you were just looking at the osteological characters. And I think, again, partially that's because we didn't know what the different groups, what the different monophyletic groups actually were. And then two, it's because the skulls themselves are physically hard to look at when you have all of these osteoderms attached to them. So one of the first things that we did to try and examine the osteology of alligator lizards was evaluate variation and quality of previously proposed apomorphic and diagnostic characters in alligator lizards. And the way that we did that was by CT scanning a bunch of them, and then looking at that sample and some previously collected osteological specimens to try and get at patterns of variation in the group. And so here are some examples of the segmented, so that's a 3D processed computed tomography renderings of some of the isolated bones from different species of alligator lizard. And what we found is that really most of the previously described characters, whatever context they were described in, the diagnostic utility of those characters was altered by intra and interspecific variation that was previously undocumented. So that was pretty interesting. And that was kind of our first pass at trying to figure out, what's going on more osteologically with this group. And I want to point out that CT was really, really critical here. Again, if you're just looking at a regular skull, it can be hard to see a lot of the internal anatomy because the osteoderms are all, covering everything up. So once we had an idea of the variation in the group and the quality of some of the previously described characters, we expanded our sample past the genera Algaria and Garanotis, and then included the two other genera, Beresia and Abronia, and created a new 80-character morphological matrix that could be used to place fossil alligator lizards and look at the phylogeny of the group. And so here's some surface renderings of the segmented skulls of all of the different groups, or most of the different groups. And in blue here, I've pulled out the osteoderms, and in that sort of silvery color, that's the actual skull. And so you can see how much it actually obscures of the anatomy that you're trying to look at. Okay, so I wanted to give a little bit of background on what the projects are and a little bit of background on the group, but now I'm going to back up a little bit for how this actually came about in the first place. So, most fossil alligator lizards are isolated cranial elements that are disarticulated from other parts of the skull or other parts of the skeleton. But there are a handful of articulated skulls that have been described, and one of these was this unidentified fossil alligator lizard that was first described by Mark Norell in 1989. And he's a pretty famous paleontologist who's now the head of fossil herps and maybe the entire paleontology division at the American Museum of Natural History. But anyways, he described this fossil alligator lizard, and basically he was unable to observe previously hypothesized apomorphies of different alligator lizard groups on the skull because the internal anatomy was obscured, in this case, not by the osteoderms, but by the matrix itself, the conglomerate matrix. And so he wasn't really able to figure out what it was. And so at some point, Chris got this specimen on loan and had it CT scanned and decided, that's probably something new. And then, he waited to have a student or students to work on it. And that ended up being me and David later on. And then another interesting thing, at some point in time, he told Harry Green that if this ended up being a new taxon, then he would, him and whoever worked on it with him would name the taxon after Harry. So I'll get to that in a sec. So the fossil is found from the Anza-Borrego Desert in Southern California, which is just a little east of San Diego. And the formations that are there span the Pliocene to the Pleistocene. So 3 to, I don't know when the youngest is, but maybe around 1 million years old. And this fossil is from the earlier parts of the deposit, around 3.2 million years. All right, so here is the volume rendering of this skull that we called Algaria Peludo Verde after Harry Green. And it's the name that he took up when he was in Costa Rica. And it was Chris's idea to give him that name. And we liked it a lot. It was a fun joke. Nice to be able to name the fossil after him. So yeah. So how did we decide that was an alligator lizard in the first place? Well, it helped that Mark had already worked on it, but I just want to lay out, how you would actually know. And the main things are that it has a non-compound cephalic osteoderms that are laterally imbricating with each other, which you can see once this flips around over here. There they are right there. So that's diagnostic of anguids. And then among anguids, having a fused frontal is diagnostic of alligator lizards. And then in terms of, how did we think, all right, this must be an Algaria? After looking at a large sample of alligator lizards and assessing the variation among them, we realized that having a bunch of pterygoid teeth, which you can't see on this GIF, and then the texture of the osteoderms was indicative of Algaria. But we weren't able to provide a phylogenetic diagnosis of the fossil just qualitatively. So we used phylogenetic analyses to try and figure out how it was related to other alligator lizards. And so I also want to talk about the quality of the fossil. So this is a rendering called a dynamic cutaway. And basically what we're looking at here is the volume rendering of the fossil itself. And then this is a modern Algaria right here. And then we're going through all of the different CT slices. So all of the different X-ray slices from the scan. And so you're seeing both the external and the internal anatomy at the same time. And even though this fossil is a little bit taphonomically deformed on the left side of the skull, it at some point got smushed in a little bit. The actual internal quality of the bones is really, really nice. And so we were able to segment it out, which is the 3D processing. We were able to do that really well. And so here's what that actually looks like. Here's all of the individual cranial elements that are color coded so that you can see them all separated from each other. And here's what the fossil skull looks like with nice macro images. And then I was just back in Austin last week, or I guess 2 weeks ago now, and David and I had the pleasure of going and herping with Harry, and so getting to celebrate the formal description of this lizard. And then here's, you can get a little bit of a closer look. Here's what what some of the individual bones look like when you pull them out from the rest of the skull. And including, here's these bones of pterygoid right here that have the pterygoid teeth on them that are one of the apomorphic features of the new taxon. Okay, so unsurprisingly, the phylogeny of Algaria has also been uncertain over the years, like the larger group of alligator lizards. And so there have been a number of different topologies proposed. Most recently, Dean Levitt, who is at San Diego State, did an analysis that explicitly tested for mitochondrial integration and proposed a new arrangement of the different species of alligator lizard, and also found potential paraphily in the species Algaria multicarinata, which is the more common one that's found across the West Coast. And then importantly, he found that the species of Algaria that are found on the Baja California Peninsula aren't all sister taxa to each other. So this species, Algaria paucicarinata, and then this species, Algaria kingi, are sister taxa instead of Algaria paucicarinata being in a group with Stadrocensis and Velasquezi, which are the other taxon on Baja California. So that was really helpful for us because I was aware that there was some uncertainty in the group, and I would have wanted to be able to constrain my phylogenetic analyses using a molecular scaffold. And so it was nice that this was published, a little bit before or around the time that I started working on this. Okay, so we did a suite of different analyses, and I'm just going to show two of them here. Is a basically fully constrained Bayesian analysis that is scaffolded to previously published molecular phylogenies, most importantly, the Levitt et al. Paper. And then there's an almost fully constrained one in which we allowed a little bit more flexibility for taxa, non-Algaria taxa that have historically had controversial relationships. And the important one here is the species Garanotus parvus. So Garanotus parvus is morphologically kind of looks like an Algaria, but our own Chris Conroy actually wrote a paper that demonstrated that it's more closely related to Garainotis. And that's been backed up by all subsequent analyses of molecular data sets. But when we allowed Garainotis parvus to go wherever it wanted in the tree, as opposed to constraining it to be in the clade, including Garainotis, Abronia, and Beresia, it was placed as a basal Algaria, which is definitely not correct, but it caused the placement of our new fossil to switch a little bit, which is pretty interesting. And so these are the two alternative hypotheses we got for the relationships of Algaria peludo verde. One is that it's a sister taxon to this group that includes Algaria multicarinata, then two of the things from Baja California, and then Algaria panamentina. And then the other hypothesis is that it's more closely related to Paucicarinata and kingi. So in either case, it's a basal, we found it to be a basal member of that clade, but those are different interpretations of the fossil and would indicate different biogeographic patterns for Algaria. So I think probably it's more consistent biogeographically with being, some basal member of this multicarinata clade. But in the modern biota, Algaria kingi isn't actually found that far from Anza-Borrego. So I think it's conceivable that it could have been a basal member of this clade if they had a slightly broader distribution at some point during the late Miocene and the Pliocene. So I'd say that either interpretation is consistent with the modern ranges. Neither is really inconsistent with the geology of the area, but it would definitely entail different interpretations of the known diversity of Algaria on the fossil record. Okay, so we'll keep that in mind. All right, now we're going to talk about everyone's favorite group of alligator lizards, Abronia. So these are really popular outside of science, actually. I'm always seeing people getting excited about Abronia on like Reddit, or I've seen them on like Instagram too, but pictures of these guys popping up because they look really cool, and scientists and non-scientists alike are really excited about them. So in the modern biota, Abronia are found in Mexico and then in Central America, and they're mostly found in cloud forests and coniferous forests and tropical deciduous forests that are restricted to mountain ranges. And so here you can see there's the principal Mexican ecosystems, and the different ecosystems on the map basically recapitulate the mountains and the range of extant Abronia. And then here's some pictures of some of these guys, some of the modern ones. That's Abronia monticola, and then that's Abronia Martin del Campoi, and then that's a species called Lythrochila. And so there've been a number of proposed phylogenetic relationships for this group as well. Again, based on osteology, there was hypothesized to be a basal split between the more dragony looking, they're called dragoncitos in Spanish, but between the more charismatic looking and arboreal taxa, and then a group that was previously termed Mesaspis, which are generally smaller, a little bit more drab, and while they still are found in montane regions, they're not arboreal. And then there's other hypotheses based on external morphology. And more recently, based on a nuclear and mitochondrial data set, there was some indication that there might be paraphyly in the group, but the relationships weren't necessarily strongly supported. And then most recently, a DDRAD analysis demonstrated even more widespread paraphyly of Abronia and what was formerly called Mesaspis than would have been previously, than I think anyone previously suspected. And so this indicates that these, arboreal and terrestrial morphotypes have shown up a bunch of different times. And I know I would like to see some research on that because I think it's really cool. There's really, either from the external morphological data or the osteological data, this was a tree that you just would never, ever get to. And there would be no reason to think that there would have been, this many independent appearances of arboreal and terrestrial taxa with these completely different morphotypes. But it's really cool. It's a really, really cool analysis, and I'm looking forward to seeing more about it, or more work on the group. And the other thing that they found was that, rather than the life history or the morphology or anything else that had been proposed dividers for the relationships among the group, they found that the phylogeny pretty closely recapitulated the biogeography of the region. Namely, that there's one group of Abronia distributed on one side of the Isthmus of Tehuantepec, and then another that's distributed on the other side, which is pretty interesting. Okay, so how did I get interested in all this? So I found this fossil when I was surveying the collections of the UCMP in 2016, so a while ago now. So I was down there looking for fossils basically to write a dissertation on, trying to figure out, what was going on with with lizards in North America during the Miocene. And so I found this fossil. I didn't know what it was. I talked to Sarah El-Shafi about it, who is in the audience today. And she said she was working on glyptosaurian anguids at the time, a different group. And she said, well, it has keels on it, and glyptosaurians don't have keels on their osteoderms. So, it really can't be a glyptosaurian. And I was like, all right, so, that's good. At least eliminated one thing. But I hadn't seen anything else that looked like this before. And so it kind of took me a while to try and figure out what it was. Again, I want to point out that it's really cool to have an articulated fossil lizard skull of an alligator lizard or anything else. So it was surprising to me that this hadn't been worked on and had just been sitting in the collection. And I guess before I go to the next slide, at some point I've started to realize that it must be an alligator lizard, again based on the morphology of the osteoderms, in addition, and then also that the morphology of the frontal, that it was fused. All right, so the fossil is from the Caliente Formation, which is a sort of late middle Miocene formation in Southern California, north of Santa Barbara. And it's inner fingers with some marine formations, which is pretty interesting. Now it's kind of a Badlands type area. And this part of Southern California is geologically really, really interesting. Obviously, I've got a couple faults here. So it's right on the San Andreas Fault and the Big Pine Fault. And then to the west over here, there's the Nacimiento Fault. And this block of rocks here, it's the southern end of the Selenian block, which is a block that has been sort of controversial in the geological literature. As recently as 30 years ago, there were some hypotheses based on the paleomag data that it might have been like a alien block of rock, essentially, that migrated up from like South America at some point during the Cenozoic, because it was I think it had something to do with the rotation, and then again the paleomag data. More recently, the sediment provenance analysis using detrital zircon found that was not the case, and that the source rocks for the sediments that are here are actually relatively local, south of the current location, which is unsurprising given that there's been some movement of this area along the San Andreas Fault, but still relatively local. All right, and so here's this fossil, again, looking at a video of the volume rendering. And I also want to point out, this is an extant Abronia right here, but it's the fossil is in two parts, and I'll show the other part in a sec, but this part is only the front half of the skull. So from the frontal and then posterior was not preserved for the most part. Okay, so like I said, again, I didn't really know what it was for a pretty long time, but once I started CT scanning Abronia for the other project, I started thinking, oh, this kind of looks like this other group of alligator lizards that I was until now unfamiliar with. And the features that were sort of indicative of that were the osteoderm texture, which is pretty different, and then the morphology of the frontonasal osteoderm, and then the proportions of some of the other internal anatomy that you can't see right here, but particularly the nasal process of the premaxilla and then the maxilla itself. And I also want to point out the name that I have given it, Abronia kuyama, is named for the region that it came from, obviously, but kuyama is derived from a Chumash word, and then the fossil was collected on Chumash land. So I think that's important to mention. All right, so again, here's one of those dynamic cutaway videos looking at the internal anatomy of the fossil. And so unfortunately, unlike the Algaria Peludo Verde fossil, while the external anatomy of Abronia kuyama is really well preserved, the internal anatomy is not. And so it was actually really difficult to do any 3D processing of the fossil because the interior of the bones has been like mostly replaced by the mudstone. And so there wasn't a lot of discrepancy between between the matrix and the bone itself. And so that made segmenting it pretty difficult, but I was able to get out some different bones, and here they are. All right, so here's a visualization of what this looks like relative to extant alligator lizards. So you can kind of put together like, all right, it's definitely not an Algaria. I don't think it's a Beresia either. And really, you know, again, guided by the osteoderm texture, especially, it really, really looks like an Abronia. But the important thing about that is that that suite of features that I talked about that seem to indicate that it's related to this group of arboreal alligator lizards is only found in the arboreal alligator lizards in the clade Abronia, which are not all of each other's closest relatives, right? So there's repeated evolution of, you know, not only the external morphological, but also osteological characters that are indicative of the arboreal groups, which are not each other's closest relatives and have appeared multiple times. So that makes it challenging to try and figure out how it's related to the rest of them, and I'll get to that in a sec. But I also wanted to show, you know, this crazy osteoderm, like really vermiculate texture you can actually see on the scales of some of the modern members, which I think is pretty cool. The other cool thing about the fossil itself, so it's preserved as a skull, and then it's preserved as this isolated block of mudstone that contains a few other bones in there, namely this quadrate bone, which is the articulation between the mandible and then the top part of the skull, and then some additional osteoderms on top of that bone. And what's really, really crazy about this is that these osteoderms have like really, really tall keels or sails, or that's what I called them. And I haven't seen this in any other alligator lizard. Really, the only lizard, only modern lizard I found this in were chordilids that have such large keels on top of their osteoderms. So, you know, as far as I know, that's an audapomorphy of a new taxon. Really, really, really cool looking morphology. Okay, so using the morphological matrix that we constructed, and then, you know, various permutations of molecular scaffolds based on previously published analyses, molecular phylogenetic analyses, you know, we tried to place this fossil. And so here, there's pretty minimal constraints. Mostly, I just constrained Algaria to be monophyletic and basal, but I allowed relationships among Abronia to vary. And then here is an almost fully constrained, or a basically fully constrained tree with respect to the molecular hypotheses. And in both of these, the fossil comes out as a sister taxon of a group of arboreal Abronia that are only found east of the Isthmus of Tehuantepec, which seems biogeographically improbable, but I'll get to that a little bit more right now. So it's become a little bit more common practice among paleontologists recently to not only try and assess the you know, morphological congruence of fossils with the osteology of extant taxa, but also to try and assess the stratigraphic congruence simultaneously. And you can do that through tip dating analyses with or without the fossilized birth death process. But in this case, I've used that as a model for this divergence time analysis. And so, you know, this is really interesting. You know, one, the divergence times actually don't look that much different from what you would get from a molecular tree, which I was a little surprised by. But then two, it shows that the fossil is stratigraphically incongruent with being sister to this group of arboreal Abronia that are found in lower Central America, which is a pretty cool result. And I think, you know, casts sufficient doubt on this fossil being a member of the crown clade that it really should be assigned to the total clade instead. And that actually makes a lot of sense because it has all these features that are common to arboreal members of the clade. But again, those individual clades aren't each other's closest relatives. And so it's kind of confounds how you would interpret a fossil that has all of those morphologies. And so in an earlier iteration of this study, I also wanted to do statistical biogeographic analyses to try and figure out, you know, how you could explain this occurrence. And so, you know, this is an old biogeography analysis that I won't publish, but it's on a combined evidence time tree that used the earlier version of the matrix and then some, you know, previously published nuclear and mitochondrial data that I got on GenBank. And so basically what I found here is that there's no real way to explain the occurrence of this fossil without invoking an improbable long distance dispersal, because there's no connectivity between, first of all, this upper Central American clade of Abronia, but really any of these clades, and then the mountains that are in coastal California. And so part of that is because, there's no Abronia in the Sierra Madre Occidental. So yeah, it was, disappointing that I couldn't, do a nice biogeography analysis that worked out well, but I think the results made sense. Yeah, I thought the results made sense. So, the hypotheses that I came up to try and answer, what I found were that, given a conserved ecology of Abronia, namely that they are living in cloud forests or, subtropical coniferous forests or deciduous forests, and that they've been living in mountains, maybe they were ancestrally occupying the Sierra Madre Occidental, and then they would have dispersed from there to mountains in coastal California at some point during the Miocene. And so in the modern biota, such a dispersal would be pretty improbable for a montane taxa like that, because there's the Gulf of Mexico or not the Gulf of Mexico, the Gulf of California and the Sonoran Desert in the way. But based on the record of fossil plants, we know that the arid environments were a little bit more localized, or a lot more localized during the Miocene. And so you didn't have a big swath of desert that would have necessarily been impassable. And then second of all, the Gulf of California really didn't start, that rifting didn't start until later on in the Miocene, and it didn't finish until the latest Miocene or the early Pliocene. And so, the timing is a little bit tight, but, given that the fossil was deposited, somewhere between 12.5 and 11 million years ago, and the rifting didn't start, somewhere around 10, and the lineage that the fossil belongs to had to have gotten to the area before 12 or 11, maybe there was no Gulf of California to migrate across at that point in time. The other thing is that, again, this part of California there has moved northwards quite a bit since the Miocene. So here's the modern day location of the Caliente Formation, and then here's the Paleo locations, which there's been a little bit of movement along the San Andreas Fault that you can see. So that's one explanation for how you could explain this record, given that this fossil appears to belong to Abronia. The other explanation is sort of a more tantalizing one, is that the ancestral range and ecology of the group was much more diffuse than you would expect based on the modern biota. And so I think, it's tantalizing because there's not really much evidence to support that yet, but maybe it's possible that the total clade was found in more diverse environments than the crown members, which are restricted to mountains and specific kinds of habitats. Ultimately, both of those hypotheses, what's really going to be needed to test them is finding more fossils. So one, I'd like to find more fossils from the area that are referable to this new taxon, to kind of confirm its presence. But then two, I think what would be really important would be to go to the Sierra Madre Occidental and try and find fossils there. Okay, so almost regardless of what this ends up being or how specifically I'm able to place it phylogenetically, this is one of the reasons that I like working on fossils, is because it provides you, it opens a door to a line of thought about biogeography or other things that you might not get from the modern biota. And another important thing that I want to mention about the distribution of modern Abronia is that we do know to some extent that they must move across areas that are not montane, because there's one group that's found, at least one group that's found on isolated volcanoes in other parts of Central America, here in Veracruz. And there's no connectivity between this set of volcanoes and any of these other mountains around here. And so, we know a little bit from the modern biota that they have to move across, non-montane areas. But so it's at least conceivable that, something could have moved long distances or relatively long distances across a non-montane area. Okay, so to sort of sum up there, limited topological changes can alter fossil placement. Repeated morphotype evolution complicates fossil placement, but then the fossils can still provide biogeographic information that's not apparent from the extant biota. All right, so now I'm going to talk about pleurodontan iguanians, another one of my favorite groups. In the modern, there's about 1,200 extant species, and I was just looking at it today, there's about 2,000 extant species of iguanians total. There's a lot of well-known taxa like horned lizards and anoles and collared lizards and things like that. And currently they're distributed for the most part in North and Central and South America, but then there's also a famous lineage that lives on Madagascar, the Uplurids, and they live in a wide variety of habitats, deserts, rainforest, scrub forest, and then sort of desert scrub interfaces, just to name a few. And so, not dissimilar to alligator lizards, there's been a number of, proposed relationships for how the different groups of pleurodontan are related to each other. So this is sort of the first pass that was actually unpublished by Etheridge, but it was actually commissioned by Ernest Williams in 1962. I think he asked, and maybe Jim would know more about this than me, but he asked Etheridge, how do you think the different groups of pleurodontan, or at that time they would be called iguanid, are related to each other? And this is the tree that he came up with. And then later on, there's, here's the first cladistic attempt to trying to figure out how pleurodontans are related to each other. And then, later on, there have been quite a few different hypotheses based on Sanger sequence, multi-locus data sets. More recently, there's been hypotheses based on sequence capture data. And I, dropped myself in there, stuck my toe in the water by trying combined evidence of sequence capture and morphological data. So lots of different hypotheses, a lot of different inconsistency between them. And even now with genomic data, there's still, it's unclear how the different groups are related to each other. Okay, so how did I get interested in that? I found this fossil in the collections of the Yale Peabody Museum when I was there for a completely different project. And I recognized that it was an iguanian, but I didn't really know what it was. It's from the Bighorn Basin around 53 million years ago. And so I got it on loan, had it CT scanned, wanted to figure out what it was. Here's a video of the volume rendering of the skull, which you can see, even though most of the cranial elements are there, they're kind of preserved out of order with each other, and there's a little bit of taphonomic deformity. And for comparison, there's a Gambelia skull right there, so you can kind of look at how the jaws are not really in correct articulation with each other, and some of these skull roofing bones are out of place. So my hypothesis is maybe this was partially digested or something before it was preserved, but I really don't know. Then here's a different kind of rendering so that you can get another look at that. And this is the surface renderings of the segmented bones that I processed, superimposed on top of a semi-transparent volume rendering of the matrix. So you can see, yeah, most of the bones are there, but some of them are kind of out of place. Okay, so again, I didn't really know what this was. And so I kind of wanted to try a number of different approaches to figure it out. So I used several of these different phylogenomic scaffolds based on different data sets that have been published recently. And then I did a wide suite of analyses to try and determine the relationship to the fossil. I used two different data sets. I used parsimony and Bayesian methods, and then I did sensitivity analyses in which this new taxon was the only fossil. And so this isn't a pretty figure in a lot of ways, but it kind of gets at the what I found, which is that the placement of the fossil was extremely contingent on the topology that I constrained the data to. So that's a pretty interesting result. And so I honed in on two alternative hypotheses using one of the matrices, which was created to determine relationships among iguanian lizards. And so here's those two different alternative hypotheses for the relationships of the new taxon on the scaffolds. So in one hand, it could be related to a group of pleurodontans that are found in South America. On the other hand, it could be closely related to protophytids and choriophanids, so collared lizards, and then basilisk lizards. And so then I did analyses, stepping stone analyses, where I constrained the relationships of the fossil to be in either one of those alternative placements, estimated the marginal likelihoods, and then did a Bayes factor comparison, and the results were equivocal. So under one topology, one hypothesis was strongly favored. Under the other one, neither one was favored, and under the other hypothesis, under the other topology, one of the hypotheses was a little bit favored. And so those are pretty interesting results, I think. So I could have, if I had just picked any one of these scaffolds and then constrained my tree to that, I may have gotten what seemed to be a more straightforward interpretation of the fossil. But because I tested a couple of different topologies, I did not. And so what does that mean for interpreting the biogeography of this fossil, for example? So on the one hand, you've got collared lizards and leopard lizards, which live in North America, and basilisk lizards, which also mostly live in North America, although there's at least one species that gets into South America. And then on the other hand, you've got these Fluorids and Myosaurids and Hoplocircids, which live mostly in South America or Madagascar. And so these are two completely different biogeographic interpretations of the fossil that would entail, some pretty different dispersal hypotheses for how a taxon related to Lyosaurids, got to be, in the northern part of the United States during the early Eocene, or, it could be more consistent with, known choriodophantid fossils that are found in North America during the Eocene, or the modern distribution of protophytids, which is in North America. So, I think that's pretty important, because those are quite different interpretations that, were completely tied or largely tied to tree topology. And I think, it's not hard to imagine that sort of problem is generalizable to fossils to other groups besides lizards, right? So, pleurodontan iguanians aren't the only group of animals on the planet that, there's some uncertainty or some controversy about how the different groups are related to each other. And so I think, it's not unreasonable to suspect that if you're to test multiple hypotheses to place your fossil, whether they're morphological or molecular or whatever, then you'll get potentially a lot of, different placements that could affect your biogeographic or ecological or whatever interpretations. So to go back to pleurodontans, this is also not the first fossil pleurodontan for which there's been a lot of uncertainty about its placement. So here's this fossil Astoprion, which has been hypothesized to be a choriodophantid, maybe a stem pleurodontan. It's North American. And then there's these really interesting fossils from the Uka Tolgad, which is a famous locality in Mongolia. And they're all from the late Cretaceous, but they've been thought to be variably stem pleurodontans or crown pleurodontans or stem iguanians, or all these different potential iterations that would have completely different biogeographic ramifications for the origin of pleurodonta and the origin of the major clades as well. So all of this kind of set in motion what I wanted to do for my postdoc. So I thought, we still don't really know how the different pleurodontans, or really, more broadly iguanian, or more broadly, all of the iguanians are related to each other. And so I thought, maybe I'll try and address this myself. So, most previous sequence capture phylogenies sampled relatively few species, and time trees, either sequence capture or previous Sanger sequence phylogenies used a few iguanian calibrations when they did divergence time analyses. And then there have been a number of biogeographic assessments of the group that have leveraged fossil data or modern data, but generally not both. So, what we're trying to do for my postdoc project is produce a densely sampled phylogeny relative to previous studies. So try and get, at least 200 of the extant species out of the total of 2,000 iguanians, using a new probe set that includes the relic markers that Ben designed that should be less susceptible to gene tree estimation error. And then hopefully with better phylogenetic resolution, we'll be able to address the iguanian biogeography. Where did all the major clades come from? What have they been doing through time? And then place fossils without worrying about excessive uncertainty, and then get more reliable divergence times. And so I'm still working on the phylogenomic part of the project, getting it going. So I've started practicing DNA extractions. I've got a lot of the tissues on loan that we're going to need for the project. And then I'm still working on probe design a little bit. but I've collected a lot of the morphological data that's going to be necessary. So around data for 50 extant and 20 extinct iguanians for this Smith matrix that was created to assess relationships among iguanians. And so now that I finally, kind of grounded myself and a little bit more settled in, I think I'm finally ready to get some undergrads to help me finish up the morphological data collection. And so although I haven't, I don't have the phylogenomic data, obviously, to start creating tree hypotheses, I've started testing out, the placement of the fossils given one of those previously published molecular trees and using the fossilized birth depth analyses that are going to be an important part of the project. And so now you can get kind of a closer look at what that looks like and my very qualitative preliminary assessment of the biogeography of iguanians, or pleurodontans specifically, namely that some of the stem taxa originate in East Asia, and then we have kind of a gradual dispersal from North America, and some clades that are North and South America, and then clades that are only found in South America or Madagascar. And I think the modern data mostly recapitulate that, and that is what has been previously proposed. But there's a lot of fun fossil exceptions, and if anyone's interested, then we can talk about that during the questions. But yeah, I just want to point out we have some stem polycrotids, stem anoles, some stem and crown choriotophanids, stem crotophytids, stem iguanids, and then some of these late Cretaceous things from Mongolia I've been getting as stem phrynosomatids, which is pretty interesting. All right, so back to what the original title of the talk. What have I learned about interpreting the fossil record from alligator lizards and iguanian lizards? So fossil placement is tied to tree topology, and that's not necessarily surprising. It makes a lot of sense, but when people use paleontological data, they don't necessarily test the tree hypothesis that they're using for their divergence time analysis or their comparative analysis or whatever it is that they're happening to do with respect to the original phylogenetic placement of the fossil. And so given that, fossils may end up in different parts of the tree depending on what you constrain the topology to, that presents a potential problem for using paleontological data. So, if you're doing your own node calibrations, you need to make sure that the fossils that you're using are placed consistently with the tree that you're actually trying to calibrate. If you're doing a biogeography analysis, same thing. This also would affect our understanding of the diversity preserved in the fossil record. So like with the fossil Copedosaurus, an interpretation of it being a basilisk lizard or a liosaur, are completely different. And that, it would, if you were trying to determine what fossils of those groups had been found by going to a database or looking at a fossil paper, a paleontology paper, those results are different. The different results. And so, really my plug here is that users of paleontological data, whether they're biologists or paleontologists or whoever, are responsible for ascertaining the, placement consistency of the fossils that you're using between the upstream and downstream phylogenetic hypotheses. So, is the tree that you're using now consistent with placement of the fossil and the tree that the paleontologist originally describing the fossil was using? All right. And so here's my acknowledgements, lots of people to thank, and lots of institutions to thank too. And now time for questions. And there's some salamanders I found last weekend for fun.
Yes, if we can thank Simon, maybe we can unmute and give him some actually auditory applause.
Yay, Simon.
I know some people may need to run since it's after 1 o'clock, but for those who are have some time to stick around, I'll let Simon address questions if people want to raise their hands, and I can watch the chat to make sure that if any questions come in there, I can relay those to Simon.
Thanks, Jim.
Well, I'm happy to start asking questions then if nobody else is chiming in. So yeah, I'm really curious about the Abronia fossil from Southern California, and and how the separation of the Baja California Peninsula from mainland Mexico may have played a role in that distribution. I mean, the Elgaria results with Posse carinata being the sister of that king-eye group. I mean, that's a pattern that's seen over and over again with Cape region fauna. They're all related to things from adjacent Mexico, and presumably because of the split, because the peninsula used to be attached to the mainland there.
For
It's
sure.
12 million years seems like a long time, so it seems like there would be some time for movement along that fault. I mean, you addressed this question in your presentation. You sort of mentioned that it probably moved about this much. You know, what I was thinking when I saw that was about this Lepidophyma skull that I think I told you about, that Jacques Odier has from Southern California.
Yep.
Which is another like tropical Mexican clade that's nowhere near Southern California now. It's clearly a Lepidophyma skull, like a really nice one. And I wonder if it would have been from sort of the same zone as Urobronia prior to plate movements.
Yeah, no, that's a great.
Something about how they got there, right? Like if Lepidophyma and Abronia both might have got there the same way.
Yeah, either through the movement of the that part of the plate up, or the terrain just being more traversable at the time. No, that's a great question, and actually, that's something that I didn't mention is that I think the interpretation of that fossil would be well supplemented by talking about other lizard fossils, or just about other groups of fossils from other groups, mammals and plants that might show the same biogeographic pattern. And so that's actually not something that I've seen a lot of discussion of, but if I have a field program in the future where I'm able to go collect, I think that's something that I would definitely want to target. But yeah, no, it's a crazy fossil. And, even before I had some idea of what it was, I was just like, this is really bizarre looking. And then once I was able to compare it to modern Abronia, I was definitely pretty floored.
Really looks like an Abronia.
Yep.
Hi, I'm Genevieve. I had a question about your postdoc plans and integrating the morphological data with the genetic data. And I guess what are your plans? You said that like getting more tips might make it more reliable, but what kind of tests are you doing? Any tests to see how consistent the fossils are in that tree, or what direction are you going with that?
Like in terms of like getting more tips to have more reliable divergence states, or
Have more reliable, say that last word again.
sorry, more reliable what?
Placement of the fossils.
Oh, of the fossils.
Yeah.
Yeah, no, that's a good point. So I mean, basically my hope is that by doing a combined evidence analysis, we'll get a more accurate placement of the fossils than occurred previously, because for the most part, that hasn't really been done. Another issue that I didn't really talk about is that a lot of previous analyses that tried to place some of these fossil iguanians were using matrices that were, designed mostly with characters to discriminate the different groups of, squamates or lepidosauromorphs in general. And so they included, mostly characters that would be useful for higher order problems. And so, the basic premise is, combining data sets using a more targeted matrix, hopefully will put them in a place, will hopefully improve the accuracy. But I think I'll probably have to think about that more. I mean, obviously it helps too that I'm like an osteology person, and I have some expertise in like the morphology of all these different things. And so I'll have some sense of whether any of these placements are unreliable or not. But to be honest, actually, none of the things that I found so far are terribly different from previous hypotheses. So I think there is some consistency with what has happened previously, which is good. I think one thing that I'm actually a little bit more worried about is that the way that I'm sampling is going to affect the divergence time analyses, because you might have noticed my sampling strategy for fossils so far is to try and sample a bunch of stem things, because I have some confidence that I could actually place those correctly, as opposed to like crown members of Anolis. Like there's a bajillion amber anoles that I, don't want to include in a tip dating analysis because I won't be able to place them accurately. And so I've been thinking about that recently, and I think I'm going to have to put more thought into, can I do like an unresolved and a resolved fossilized birth analysis at once, which I haven't seen people do, but maybe I don't know if you have any thoughts on that.
I definitely would be down to chat sometime and think about it.
Yeah.
I don't have any thoughts off the top of my head right now, but it's something I've been planning to look into.
Oh, great. Yeah, no, I'd love to chat about it. Yeah, no, those kind of analyses are a little new to me right now. I've done lots of, tip dating without the fossilization process, and then, lots of node dating analyses, but I haven't done so much FBD. So yeah, definitely down to talk shop.
All right, unless there are final questions for Simon, I think we might just want to thank him again for a great and definitely a thought-provoking talk about phylogenetics and fossils. I've loved the CT scans, by the way.